Welcome back, friends. In this video tutorial we'll be talking about a bacteriophage lambda, and we'll be talking about different aspects of bacteriophage lambda, the structure of bacteriophage lambda, the genome orientation and genome structure, and different genes that are responsible for converting the lambda lytic and lysogenic cycle, which is very important topic. Okay, now previously we have already talked about that lytic lysogenic switching, so in this video we'll be not talking about that. We'll be talking about how the lytic cycle actually established and how lysogenic cycle. So let's begin with the structure. Now lambda is a bacteriophage because it infects a bacteria, right? So it's a virus that infects bacteria. Now in this lambda is belonging to the family Siphoviridae because their structure resembling the structure like siphon or siphon, we call them the Siphoviridae. So let me take the color here. Okay, so we call them Siphoviridae, and the host for this lambda phage is K12 strain of Escherichia coli. Now E. coli are having many different strains. Among them, K12 is very important strain, and this lambda is infecting those K12 strain of E. coli. Now if we look at the structure of lambda phage, what we can see here, this is an electron micrograph. This is also electron micrograph of lambda phage. Now in this case we can see that there are three important regions for this lambda. One is this head, so this is the head region of lambda phage. Second one is the tail region of lambda phage, and third is called the J chain at the end, right? It's called the attachment fiber or tail fiber. It's called the tail fiber, or simply called as J chain of lambda. Now this is the chain which is important. It will something look like this. So if I draw it schematically, how it will be represented, it will be represented like that. So let's say this is, and the J chain will something look like this. Okay, and then the icosahedral head will be aligned like that. Okay, so that's how the lambda actually looks like. And the lambda phage is not having any kind of base plates or spikes and all these things that you can see in many of the images that you're going to find in your book and internet, because those type of phages are different. Those are called usually T4 phages and other type of phages, but lambda phages are not having those base plate or this that kind of spikes. In turn, they are having a kind of tail fiber which is required for their attachment, right, or adherence to the host cell. And they are having this kind of contractile tail, non-contractile tail, sorry. Now there's a difference between this lambda phase and T4 phase. If I am talking about in my YouTube channel, you can find videos and structure about both lambda phase as well as T4 phase. And you can find this in T4 phase. What we can see, that tail region is kind of contractile and it behaves like injection. So this tail region, if this is the length of the tail, it can be contracted to this small amount, and due to this contraction they can inject the DNA or genetic material inside the host cell, right? Now but in case of lambda phage, the tail that they are made up of, this is made up with non-contractile rings. Okay, so they cannot contract, but instead what they can do is that they can push the DNA inside. And but this tail region is a little bit longer in case of this lambda, whereas in case of T4 this tail region is a little bit shorter. Okay, now the lambda tail region is little bit more flexible in nature. So you can see in these images they are kind of bending and moving, and all these things can be done. So it's bended like that, something like that. So they are pretty much flexible, and more flexible than the T4 phage, T4 phage contractile ring. Okay, and this tail is actually made up with two important stages, two important organization. One is the circular rings, and inside you are having a hollow chamber, right? So if you look at the discovery of the bacteriophage lambda, it was by Esther Lederberg, and here is Joshua Lederberg. So actually regarding this lambda biology and lambda molecular biology, these two person are providing most of the important uh, what you can say experimental results. Okay, so if we look at here, how they have proved that there is a bacteriophage, which is a virus which can enter and destroy the host bacterial cell. So what they have done that, firstly they take the donor cell which is E. coli K12. Now they took this E. coli, and what they put there, they also put another recipient cell which is also E. coli, right? So E. coli and E. coli, and what they allow here in this case, they allow that these two cells to be conjugated with each other by the pilus, right? So after the conjugation, what they found that this new recipient cell simply get lysed. Okay, previously the donor cell is not getting lysed, but now after this conjugation, this recipient cell is getting lysed. Now in second experiment, what they have done, that they take only donor cells, and what they put here, they put little bit UV there. After some time, what they found, that they are again getting lysed. But when they put this donor cell as it is, without the treatment of UV or without any kind of conjugation, this donor cell remains as it is. It is not being dead. But now if we put some other recipient cell, and after the conjugation it transfers, that means it transfers something to this recipient cell, which causes the recipient cell to die by the lysis. On the other hand, when we put UV radiation or UV radiation to this donor E. coli cell, this UV is triggering something inside the E. coli cell, which is also telling the cell to die by the lysis, right? So that means there is something inside this donor cell which is a kind of dormant, for certain time. When we put UV, it is getting activated, and it is getting that's why it is getting killed by lysis. Or sometimes when they are transferring via the conjugation tube between this donor to recipient bacteria, in that case it is also providing that that factor into this recipient cell, so that the recipient cell is can be dead, right? So that's the reason, that there is something inside this donor cell. And later it was found that this thing which is present in donor cell is definitely a part of some foreign particle, which is called the virus, we all know. And this virus is called the bacteria eating virus or bacterializing virus. It is called bacteriophage. Okay. Now among them, they term this name as lambda or lambda phage in this case. Okay, now both these lysis can be studied under the microscope, and when they found this under the microscope, they found that there is something important particle that is kind of disrupting it, right? Now for the dormant state, in case of this donor E. coli, it is due to some kind of incorporation of the viral particle or the viral genetic material with the host. And nowadays we know that this dormant living of the phage particle inside the E. coli cell or inside the host cell is termed as lysogenic cycle, right? We know that it is termed as lysogenic cycle. And when they have encountered some stress, like in the form of UV in this case, this lysogenic cycle can be converted into the lytic phage, and those phage will lyse the cell, right? So after the cell lysis, usually how the experiments are conducted, that they take the plate and they put the bacterial on there, and then put this phage culture. You can see where the phage is lysing the bacterial cell, the cells are kind of dead, and they form this kind of transparent regions which are called plaques. Okay, so this is called plaques or plaque forming units or PFUs. Now life cycle of temperate bacteriophage. Okay. Now temperate bacteriophage means in this kind of bacteriophage are having both the type of life cycle, either lytic or lysogenic, both mode of life cycle here. Now in lytic cycle, what their actual goal is, that the bacterial actual phage goal is to replicate inside the host cell and then lyse the host cell and come up with all of their progeny viruses, right? But in case of lysogenic cycle, what their aim is to put their genetic or genome, put their viral genome inside somewhere inside the host genome or the bacterial genome for certain period of time. After that, when they turn the environment is turned opposite to them, then this lysogen will be converted to the lytic phage, and then they again lyse the cell and then can come out, right? So if they want to stay for longer period of time inside the host cell, they will choose lysogenic cycle. If they want to just replicate inside the host cell, they will choose lytic replication cycle. Okay, now there is a switch, very important molecular switch to select from lytic or lysogenic cycle between their living. Okay, and we'll be seeing what are those switches to find that. Okay, but usually what happens, say this virus enters and it delivers their genome. Usually the genome for lambda is DNA. Now the DNA is then getting transcribed, then replicated into proteins, then they finally organize the proteins, and then the phage particles will come out by the lytic cycle, right? But for the lysogenic cycle, they can incorporate their genome with the host bacterial DNA or bacterial genome, and it can stay there for several times. Okay. Now one important thing I must tell you is that nowadays most of them, in fact all of them, phage particle or lambda phage particle we are working with are a variety called Lambda papa. Now what is Lambda papa here? Now Lambda papa is simply derived from two important individual lambda type, which one is Lambda Paris, another one is Lambda Pasadena. Uh, that name suggests all that lambda is from this Paris. A lambda phage are obtained in Paris and the lambda phage obtained in Pasadena. And they cross these two and they finally produce this lambda papa, which is designated as a model lambda phage to study rest of the processes nowadays. Okay, okay, where is it? Okay, so let's move on.